Hello students, welcome to my channel. In this class, we will learn about modification interference assay footprinting. What we have done just recent before this, we done modification protection. See how the modification interference is different from the modification protection. So, modification interference is based on the principle that alternation of nucleotide critical for protein binding can prevent protein binding. And how we alter this? By the addition of methyl group. What this line is saying, it is all that define the difference between interference and protection. Here we just on this DNA strand, we just alter. What we alter or change? Alter means change. What we change? We change some specific nucleotide sequence which is specific to the binding protein, but we are changing it. How we are changing it? By addition of methyl group. Let's see, or let's suppose this region where A, C, T, G is present. So here what we do, methyl group will add in this position. So now with the addition of methyl group, this whole sequence is now restricted or prevented from the protein binding. But if there is no methyl group, so this whole DNA sequence or this much sequence, other part is not specific for the protein binding. This is only the part where protein can bind. But this is also changed or not have ability to bind that protein because of the methyl group is there to protect or prevent the protein binding. So this is how protection or interference. We are what here in why it is interference? Because here we changing and we interfering with the DNA sequence. So the protein which is protecting from the cleavage of that sequence from a protein which is not able to bind there. So now let's see once again. In protection, what we do, we take DNA and we have protein, and which is preventing the protein from binding of the methyl group. Here we are doing, this is DNA, we are binding methyl group, so we are protecting from the binding of protein. So we are just changing this position. First, this protein is what is doing? Protein is protecting from the modification. Here what we are doing? We are just interfere between protein and DNA. That modification we done, which is interfere, which is preventing, inhibiting the protein to bind the DNA sequence. This is the only difference or all the difference from the modification protection to the modification interference. So as this word can define, it is interfering between what? DNA and protein. In this assay, DNA is exposed to modifying agent. So what we are doing, taking DNA sequence and then we exposing it or treating with the agent which can modify or add methyl group to that DNA before complexing with protein. Before to what? Before binding that DNA with the protein. The subsequent binding reaction produces two population. So this is how two population will create one free DNA. Free DNA from what? Protein binding, which is free from the protein, and DNA bound to protein. Which DNA will bound to the protein? Where modification is not done or interference is not done, or that particular sequence will unable to have modification or interference. This population are defined by whether modification interference with protein binding. So these two population will define where the interference is exactly happening. How? Let's see again. Let's see this these are the DNA sequences. See with the focus, just see. You will clear all doubts. So if we see, this is G sequence here in this DNA sequence, this, all have this much of the G nucleotide in the DNA sequences. Okay, and we just allowing, same as the protein protection modification, we just allowing single site where we do interference modification. Let's see, in this sequence we do modification here, in this sequence we do here, in this here, in this here, in this here, and if there is some more, then let's just understand with these only. Okay, so if there is a modification in those places and all strand have only single modification, so there will be chances that have different site of the modification. So if some protein only bind to this site, so if there is modification, so this DNA is still have the protein. Okay, if this modification is here, then protein is still binding here, but in this there will be absence of protein. In this there is presence, in this there is presence. So if we are doing only single place where modification, so single place is this only. Okay, then we can find out the exact sequence where the protein is binding with this type of modification. Okay,